Hello and welcome to dnalogy.net. Today's tutorial is about genes and alleles. So here we'll just talk about the basic difference between the genes and the alleles. So let me start this by quickly telling you the structure of the DNA, which we already discussed. This is just to give you a quick recap as to what we did in the previous videos. So if you remember, the structure of the DNA is double helical, which looks something like this. So there's a double helical DNA. Now, just for convenience sake, let's try to unwind the DNA molecule, and instead of making this DNA molecule look more twisted, let's just have the simple two strands of the DNA molecule like this. But then originally, this is what the orientation of the DNA molecule is. Just to understand the concept, we will unwind the DNA and we will try to look into it. Okay, so these are my two strands of the DNA molecule, and of course, here we have our nucleotide bases on it, the nitrogenous bases on it. So let me just complete that for you here. Now, assuming on one strand of the DNA, let's say we have A, T, T, G, G, and C. So on the other strand of the DNA, we will have complementary nitrogenous bases. So for A, the complementary nitrogenous base is going to be T, and for T, it's going to be A. For T again, it's going to be A. For G, the complementary base would be C, and again, we'll have a C over here, and finally a G. And if you remember, between A and T, there are two hydrogen bonds present. Between G and C, there are three hydrogen bonds present. So this is what we did in the previous videos on the structure of the DNA. So we'll go from there now. So if this is the arrangement of the DNA inside the cell, then what exactly is meant by a gene? Now gene is, so let me just write that here, some comments for you. Genes, first of all, these are the functional units for a chromosome or a DNA. So these are the functional units for DNA. Okay, now what is meant by that? What exactly do you understand by functional units? So functional unit is something which is the smallest entity for the structure of the DNA. So if you understand the function of this basic entity, which is a gene, you can probably understand the entire structure and the function of the DNA. So it's just the smallest unit. For example, we say the basic unit of life is a cell, which means if you study the structure, the function, and the inside of the cell, you can probably understand everything about the life. So that is what is just meant by the functional unit. So for a DNA, for a chromosome, the functional unit is referred to as a gene. So why is this known as a functional unit? Definitely it performs a very important function for the DNA molecule. So what is that? So as per the definition of the gene, it is a transcriptionally active unit. So let's just write that down here. Transcriptionally active unit, which means that this specific entity, which is a DNA, is capable of transcribing into a protein and ultimately forming a protein. So since this particular function of the gene is to produce a protein molecule, a functional molecule, it is referred to as an active unit. I would rather say an active functional unit for a DNA molecule. So that's what a gene is all about. So if this is the entire stretch of the DNA molecule, it's not that the entire chromatin or the entire genetic information which is present inside the cell is functional or is completely active with reference to the protein function. No, it's not like that. So I mean to say, if I have a cell inside which there is a nucleus, and inside the nucleus we have our chromatin material present, which is definitely in the form of DNA. So the whole of this DNA, the whole of this chromatin is not active. Maybe a part of it is active and the remaining part is just inactive. Now, as per the studies, it says, of course, the 100% of the genome is not active. Out of this, a very low percentage, I would say maybe just 2 to 4% of the DNA is what is active, and the remaining is, so I would say probably 98% of the DNA is just inactive. So what is meant by this active and inactive? I just mentioned over here, active is something which is capable of transcribing into a protein or forming a protein, whereas inactive is something which is just not capable of forming any protein. It's just junk. And for that matter, this is also commonly referred to as junk DNA, which is not doing anything, but then this is just sitting inside. Okay, now we have some specific terms which are assigned to the active and the inactive part of the DNA. So the part of the DNA, or the part of the chromatin which is active, is referred to as, let me just change the color. The part of the DNA which is active is referred to as euchromatin. Sorry, this is M. Okay, euchromatin. Whereas the part of the DNA which remains inactive, or what we say as the junk DNA, is known as heterochromatin. Okay, so heterochromatin. So the active portion of the DNA is euchromatin, and the inactive portion of the DNA, or the junk DNA, is just heterochromatin. Alright, so going back to our definition for the gene, so I just mentioned gene is a functional unit. It's a transcriptionally active unit which is present on the DNA. Now this unit, since this is active, this is definitely a component of the euchromatin. A gene could not be seen on the heterochromatin area because this is a junk DNA. So it's just a dominant DNA which is not participating in any kind of protein synthesis. It's only the euchromatin which is involved in the process. So quite obvious that genes would definitely be located over here in the euchromatin area. So again, I would like to go back to my structure of the DNA, which is present here. So if this is the DNA and if this is the arrangement of the nitrogenous bases, then part of it could be active. Let's say this is the active portion of the DNA. So this active portion is our euchromatin region, and this is considered as the heterochromatin, or just the junk DNA. So on the DNA, let me just again draw that here, maybe a big one this time. So this is the DNA, and of course we have our arrangement of the nitrogenous bases. Could be anything, and we have complementary bases on the other strand as well. So part of the DNA which is active is a gene. So on that basis, we can add one more point to our description for the gene, which could be referred to as, like, a gene is approximately 3 base pairs up to, let's say, a couple of thousand base pairs. So this is approximately the length of the gene. A gene could be a minimum of, like, let's say 3 nitrogenous base pairs, and it can go up to maximum of 1000 base pairs. So that could be a gene. And a gene will be a functional unit present on the DNA molecule, which is capable of entering into the process known as transcription and ultimately synthesizing proteins. So that is a gene. So I believe now we understand what is meant by gene. On that note, let me explain you what is an allele now. Alright, so I'll go back to my structure of the cell. Maybe I can draw that here once again for you. So that's the structure for the cell. Inside we have the nucleus. Okay, now human beings, they have 2 types of cell. So cells could be either somatic, or cells could be known as the germ cells. So somatic cells are just the normal body cells, whereas the germ cells are also referred to as the sex cells. Now we, the human beings, we have altogether 23 pairs of chromosomes. Okay, 23 pairs of chromosomes. which means out of these 23 pairs of the chromosomes, 22 pairs are 20, 22 pairs are just referred to as autosomes, which means these are the chromosomes which are not participating in any reproduction process as such. But then we have this one pair which is involved in reproduction, or these are just pair of sex chromosomes, and these are referred to as allosomes. Now, allosomes could be of two types, which is for female and male. Okay, so in case of males, oops, I'm sorry about that. Okay, right here. In case of males, the total pair of allosomes are X. Sorry, males are here. It's X and Y, whereas in case of females, it's X and X. So XX in case of females and XY in case of males. So altogether, it could be written as 22 pairs of autosomes plus one pair of the sex chromosome or the allosome, which could be either XX or XY. Now, if you notice, I'm constantly saying these are pairs of chromosomes. So that means if I have chromosome number 1, it would exist in the form of a pair. So this entire bundle over here is referred to as the first pair of chromosome. So we have these two chromosomes which are present inside. That's the first pair of chromosome. Okay, so what is meant by this? If we try to zoom into the structure, we will see there is one chromosome, and then we have its counterpart, which is this another chromosome. Now both are considered as the first pair of the chromosome, where we have these two being called as the homologous chromosomes. So these are known as homologous chromosomes. Now there's a very important meaning with reference to these homologous chromosomes. Homo means same, and logos refers to the structure for the chromosome. So these two chromosomes which belong to a pair, they completely resemble with reference to their structure, with reference to their functions. So if we, let me just scroll my screen down. So if we look at the first chromosome, let's draw that big here. That's the first chromosome. And its homologous chromosome, this time I'm drawing with a different color just to have the concept clear for genes and alleles. Oops, it looks a little distorted, but it's good enough to understand the concept. All right, so on the first chromosome here, we can have a gene, and I definitely want you to remember the characteristics, the points that we discussed about the gene. So a gene could be the functional unit, a transcriptionally active unit, which is capable of doing protein synthesis. And this is present on the one of the chromosomes of the pair. Now, on the other side, on its homologous chromosome, we can have the counterpart of the same gene exactly at the same location. Or I can say the address of these two genes is exactly the same on these homologous chromosomes. And these points, so these locations for the genes, are known as loci. Okay, so the address or the loci of this gene would be exactly similar to the address or the loci of the other gene on its homologous chromosome. So if we consider this to be a gene, then on its homologous chromosome, the counterpart would be considered as an allele. So this would be the allele, and this can be referred to as a gene. On that note, we have a definition for the allele, which could be referred to as nothing but a specific variation of a gene. A specific variation of the gene. And that is what the allele is. So technically, there's not much of a difference between a gene and its allele with reference to the function, with reference to the location. It's just that these are just complementary to each other. Okay, so with that information on genes and alleles, we are now in a position to understand another two important terminologies, which is the dominant gene and the recessive gene. Okay, so that's the beauty about these genes and alleles. Now, genes, let's first look at the genes. Gene could be either dominant or recessive. So, if we assume that the gene over here is represented by, let's say, this big H, so this would be a dominant condition or a dominant gene, which is represented by a big H, and its counterpart or its allele could be recessive in nature, and that would be represented by a small h. So any gene or any allele which is dominant is always written as the uppercase, whereas its allele is always written in its lowercase. So that's dominant and recessive. And as the name suggests, dominant genes, these are the ones which express themselves, or the phenotype, the physical appearance, is just based on the presence of the dominant gene. And the recessive, on the other hand, as the name suggests, these are the genes or could be alleles which do not express themselves completely in presence of a dominant gene. So if I have, again, if I go back to my homologous chromosome, so if this gene is dominant, that would be written as a big H. And even if the allele happens to be a dominant one, even that is written as a big H then. In that case, the genetic makeup of this specific chromosome or pair of chromosome would be H and H, which is referred to as homozygous dominant condition. Reason why this is known as homozygous, because we have the same dominant gene present on both the homologous chromosomes. So it's homozygous dominant. Instead of this, let's say if we have H and small h, then in that case the condition is no longer homozygous, it's no longer same for both the homologous chromosome. On one chromosome we have a dominant one, on the other one we have a recessive one. In that case, this condition is represented as heterozygous condition. Heterozygous condition. And here you need not write dominant or recessive, because just the word heterozygous by itself explains the presence of one dominant and one recessive. We have another condition where you can have both the genes or both the alleles as recessive, so there is no dominant gene present at all. In this case, the condition is referred to as homozygous recessive. So this is just to explain you that there are certain genes which express themselves only in the homozygous recessive state, which means if one of the gene is dominant or if one of the allele is dominant, the recessive one would not express itself. However, if both the genes or both the alleles are recessive, in that condition, the specific trait is definitely expressed. So these are the three genotypes that we have for genes and alleles, and this is the basic difference between the genes and alleles. So just to give you a quick recap, like a quick definition for genes, these are the transcriptionally active unit which are present on the DNA, and the counterpart of these genes on the homologous chromosome at the same loci would be the alleles here, and the alleles are nothing but a specific variation of its counterpart gene. So that video explains you the basic difference between these two entities, and with this information we are in a position to discuss the more complicated aspects of the genes and alleles probably in the next video. Till then, have fun, see you in the next one, take care, bye.